Hello, everyone. Welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Solutions for Viral Vectors, Enabling Platform Approaches to Advance Cell and Gene Therapies. Cell and gene therapies represent two of the most promising areas in life science R&D, both from a novel therapeutic perspective and from a financial point of view. For example, Global Market Insights estimates that the cancer gene therapy market, which totaled $805.5 million in 2015, will grow to $4.3 billion by 2024. Viral vectors will play an essential role in this and a wide range of other cell and gene therapy applications. New upstream-based technologies, such as suspension production kits for high-density viral vector production, can provide for the efficient and economic manufacture of these critical viral vectors. And this is the key topic for discussion during our webinar today. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. But first, let's meet our webinar panelists. Dr. Xavier Dumoulin-Dujeu is Director of R&D Transfection at Thermo Fisher Scientific. Stephen Soltis works as Senior Field Application Scientist, also at Thermo Fisher Scientific. Xavier and Stephen are going to describe viral vector production workflows and how Thermo Fisher Scientific's viral vector solutions can optimize platform approaches for advancing cell and gene therapies. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many questions as possible. I'll now turn the webinar over to Stephen Soltis, who will shortly introduce Xavier Dumoulin-Dujeu. Stephen?
Thank you, John. This webinar is for the Solutions for Viral Vectors, Enabling Platform Approaches to Advance Cell and Gene Therapies. You have most likely noticed an increased interest in cell and gene therapy within the pharmaceutical industry, with many larger pharmaceuticals starting cell and gene therapy divisions and smaller pharmaceutical companies specializing in cell and gene therapy emerging with more frequency than ever before. This increased interest has spurred the demand for manufacturing processes that are comparable to existing large-scale processes, unlike those for manufacturing antibodies. The difficulty being that this is a relatively young industry, and these therapies were created in academic laboratories. Many batches suitable in scale for a small population of patients. These processes must maintain biological activity, ensure stable transgene expression, prevent transmission of infectious diseases, while complying with strict regulatory guidelines. Thus, the manufacturing of those therapies pose a unique challenge to the industry. You have to create a large-scale manufacturing process that is inexpensive, yet maintains the stability and activity of the viral vector. Xavier and I will go through some of those solutions that Thermo Fisher Scientific has developed for both upstream and downstream processes of these viral vectors in large-scale manufacturing.
All right, thank you, Stephen. So let me start this webinar with really talking about upstream process, meaning the process of producing the viral vectors. And really our goal here at Thermo Fisher and within my group is really to improve this process, make it more efficient, which overall will reduce the cost of producing viral vectors and make those therapy possible to go on the market. And specifically, I'll focus a little bit on the gene-modified T-cell workflow, as you can see here on this slide, where vector productions play a major role in that workflow, in which you're trying to transduce T-cells that would express a receptor, for example. And so really, today I will focus the portions around the vector productions and what system we've developed to really improve the lentiviral productions. And specifically, there's different ways to produce lentiviral. Typically, people use packaging cell lines, 293s, and they can be in two different formats. They can be an adherent, meaning they are sitting at the bottom of a dish, and then you transfect the cells. You do a transient transfection on those cells to produce the lentiviral. So that's adherent. it works well, but it's amenable for research use, meaning it's small scale and easy to use. However, when you try to scale this up to get a lot more lentivirus, it becomes very cumbersome because there's a lot of, for example, you have to use the cell factory, which is those plate stacker that can be very heavy and hard to handle and really difficult and also expensive. So what we try to do is first try to improve this process of adherent culture by switching the transfection reagents, and at least you may not fix the problem of adherent in cell culture, but at least you will get better yield, so overall we reduce the cost. But really, and I will show you later in later slides, we really try to develop new next-generation lentiviral solutions around using suspension cells. And the idea with suspension cells is really provide more scalability. But let me start first with the adherent system, in which what we observe is by switching the transfection reagents, a lot of people are using, for example, PEI, polyethylenediamine, for this. But we noticed over the years, in my group, we developed new transfection reagents for different applications, such as lipofectamine 3000, for example. We noticed that you can actually use these reagents to produce lentiviral vectors. And what you see here on this slide is an experiment in which we use adherent tuna-3 cells, and then we transfected them with different transfection reagents, whether it's PEI, lipofectamine 2000, or lipofectamine 3000. And as you can see, by just switching the transfection reagents, obviously you get better transfection efficiency in those cells, you get better production of the protein, which results with higher production, higher titers, higher production of viruses, and therefore you can see a difference of sometimes tenfold difference. So that will drastically improve your yield, reduce your cost for lentiviral productions. And I really won't talk too much about this today. The focus is really around suspension cultures and large scales, but if you want more information, you can click on the little URL on the right here. We develop an app note that really explaining the protocols, what we've done, and showing that switching the transfection reagent to lipofectamine 3000, for example, can make a drastic effect on lentiviral productions. But let me focus now really kind of what we think is going to be the next generations and where people want to go is really the ability to produce large-scale lentiviral or over vectors, real AAV. And the idea is we wanted to develop a system that gives high titer. The reason for this is because a lot of lentivirus will be used for cell therapy, for example, and it's important to increase the high titer, mostly to reduce the costs because it's still very costly. In developing, for example, T-cell therapeutics, 40% of the cost is really dedicated to the production of the vector, and that's going to be a problem to commercialize or to develop those molecules. The goal is really to reduce the overall cost of those therapies. So by improving the titers, that will allow you to use smaller volume, smaller tank, and really kind of reduce your cost. We want the system to be scalable. There's going to be a lot of research being done at small scale, and that research needs to be scaled up to larger volume, 100 liters, 200 liters. So it's very important, and this is where suspension culture really fits very well with that need. And then finally, we want all our products to be GMP. The idea is you can use those products at the research level, but you want to take them all the way toward your clinical trials, so phase 1, phase 2. So we want to provide to you the best possible reagents that are GMP. That's kind of the three major pillars of these programs. So what I'm going to show you now is really the idea behind this program. What we want to develop is a system in which when they work together, it will give you the maximum yield of viral production. And here what you see is a description of the process of making lentiviruses. So it's still a transient transfection process in which you transiently transfect vectors, packaging vectors, such as viral-powered lentiviral packaging mix that you can see here, with your gene of interest. So it's a 4-plasmid system. You transfect them in a packaging cells. Typically, there are 293 derives. And then after transfections, 2 days later, you can collect the lentivirus and ready to go. And then you go further down on the purifications, which Stephen will show you. what we want to do is really improve each step of that process. We want to have the best transfections. Those cells in suspensions, they grow in very high density with small volume, small cell surface, so they can be difficult to transfect. We want to develop new reagent for this. We want to develop new media supplement that allow us to control the cell growth so you can actually optimize really the productions of the cells. We want to use cells that can grow in very high density without clogging, without dying, to really maximize the productions. And finally, we also want to optimize the enhancers that have been shown to really improve LV productions. And this together will form the system that we want to develop to really improve LV production. And so the idea is, like I said, it will be a system. So I should mention this system is not available yet. It will be launched actually mid-next year. But it will be composed of multiple components. One is the media supplement and media that allow to sustain the growth of the cells in high density. The supplement also will allow to control the growth at the time of transfections. We have high density proprietary cells that will be used. They're very performant. They can grow in very high density and produce a lot of viral vectors. We will also develop a new transfection reagents. A lot of the transfection reagents on the market that we develop in the group are actually a lot for adherent cells. So suspension cells is a little bit more difficult because those cells tend to be smaller, so more difficult to access with transfection reagents. And also, we're trying to transfect 4 different plasmids, which also kind of raise the level of difficulties for transfection. So we really want to kind of develop the best transfection for that specific applications, and then also an answer that will really boost the LV. And the idea is really, at the end, you will produce a lot more lentiviruses, sometimes 20 to 30-fold better. So imagine in a production mode, when you want to do, for example, 100-liter scale, well, by improving by tenfold, just tenfold, it will allow you to get the same amount of virus with 10 liter, so drastically reducing the cost of producing lentiviruses. Now, like I said, this product is not available yet. You can't on our website, but it's available through beta testing. So if you're interested in producing larger scale, clinical grade lentiviral vectors, then please contact this email here, outlicensing@thermofisher.com, and we'll be happy to set up a collaboration or evaluation with you. Now, what I want to do is kind of walk you through the process of what we do in the lab, of how we discover those reagents for the system. And typically, so we do a lot of design of experiments. There's a lot of parameters that we're looking at. Again, it's a system, so each ingredients have been optimized to work with each other. So there's a lot of optimizations around what's the right cell density, which media is compatible with transfections, what supplement we should use to control the growth, the ratio of vectors, the ratio of reagents to each other. There's a lot of optimization, and this is where design of experiment is very useful. Now, even though we want to develop for large scale, all the screening was actually done in a smaller scale, in a 1 ml deep well 96, to really allow us to test a lot of iterations of the system. Once we define the leads, we identify the key components of the system, then we want to verify and slowly going up in the scales from 30 mils to bigger flask. And ideally, at the end, really kind of develop protocols that will be adaptable to larger systems like the wave bioreactors or bioreactors. So that's kind of the workflow, the process of this product. Now, I want to show you kind of each component of the system. And it really starts with the cells. It's very important to have cells that can grow in high density. And here you see a curve of the cell growth of those curves. And you could see they can go in very high density. For example, at day 6, day 7, you see density of 10 to 12 million per ml. That's very high. And also, you can see in the little graph, you can see the viability is not affected. So we have cells that can really support high density, that don't die. And so it's very important for high productions. And also do not aggregate, which is typically a major issue when you try to grow larger scale high density cells. So let's start with the cells. We're good there. Now moving on to the next aspect is You want your cells to grow. Obviously, the more cells you have, the more they can produce, but you got to be careful because you don't want your cells to grow too fast because they will spend a lot of time trying to multiply and grow and spend less effort in producing whatever you're looking for. It could be a protein, but in this case, it would be a viral vector. So what you want to do is kind of control the growth, and the way we do this is through supplements, through food. You can actually control the growth. And what you see here on the upper left is different addition of a supplement that can actually slow down the growth of those cells. And on the right is what happened when you slow down the growth of those cells and what happened to the titers at the end of the day. And you could see if you let those cells grow very fast, it will actually affect your titer that you see here on the right. But if you slow them down too much, which is the extreme right, you see also will affect your titer. So you want to find a fine balance and really kind of not have them growing too fast, not too slow, to really maximize the titering. And this is here an example of finding the right supplement. So we have the cells, we have the supplement to control the growth. The next step is really finding the new transfection reagents. And at Thermo Fisher, we have a lot of experience in transfection reagents. Actually, the first transfection reagent we launched was lipofectin in 1987. So since that time, we really collected a large library of compounds or lipids really that help with transfections. And what we wanted to do is find something that works specifically for these applications. And here, this is an example of what we do in the lab, a typical screen. Again, this is really kind of the final graph. There's a lot more upfront work that we're done here, but this is kind of showing the different candidates and how those different candidates can affect drastically the titers, the production of lentivirus. So you could see we have different candidates that really kind of improve the production of lentivirus. So again, cells, supplements, transfections. The next step is really enhancers. And there's quite a bit of enhancers that have been reported and developed. And what we wanted to do here is really kind of an exhaustive list, an exhaustive screen of what are the best molecule, what concentration, what ratio, combinations that really would improve lentiviral productions. Again, this is an example of a screen here where you could see enhancer can make a drastic difference and really improve, sometimes tenfold, the production of lentiviruses, as you can see. I should mention I show a lot of titers on lentivirus. I know there's a lot of questions and there's a lot of different ways to titers. The way we do our titers is the functional titers, in which we do transient transfection in the cells, and we collect the supernatant in the media, and then we do serial dilutions, transfect HT1080 cells, and then measure the positive cells, the GFP cells, and then that indicate the titers. So I should mention the titer that you see on the left here are undiluted LV, and again, functional titer. So you can see really, really high titers in a 1 to 2, 10 to the 8, as you can see here. And so the idea is now we develop a protocol. So now we have each component of the kits. Is how do you put them together? And protocol is actually very important as well. And here, this is an example of what the protocol will be. Basically, you would want to seed your cells at 3 million per mil. The next day, you would add the supplements. So basically, you would slow down the growth, and then at the same time, you do the transfection. So you mix your DNA with transfection reagents, incubate 10 minutes, add to the cells. Then 16 hours later, transfections, you would add your enhancer, and then 48 hours later, you can collect the supernatant, and then you have your lentivirus. Then Stephen will show you the process after this. How do we purify those vectors? But here, I'll just show you the production process. You can see very, very simple, few steps, and really maximizing the titer. Now, again, like I said, this is really still a project, ongoing project. We have a beta model that you can test for beta testing kits, but this is kind of showing our progress. And you could see here on the left, the blue bar is kind of what's being reported in the literature. People have I've developed suspension system using the PEI transfections and kind of give you the titer here, which is in the mid 10 to the 7. There's also electroporation system. So electroporations, the advantage is kind of give you really good transfection efficiency, but the problem is very cumbersome and you have to use electroporation device. It's really not very amenable for large scales, but it kind of give you an indication of what you can achieve if you have a very good transfection. So you could see here it's been reported in the 110 to the 8. When we started this project, which is generation 0, using kind of existing products, that were developed for protein production, for example, we were in the mid 10 to the 7. As you can see, generation through multiple generation, 1, 2, 3, 4, which is really optimization of the protocol, developing those new cells, developing those new supplement, LV and answer. Now we can achieve 5 to 6, 10 to the 8 titer. Again, this is undiluted titer. This is directly taken from the media, and then it's functional titer on T80. So it's a really high titer, and overall, which overall will reduce the cost of production of titers. So I should mention those titers. This is done in 96. When we scale it up to 30 mils or 400 mil, we actually have a little bit lower titer, more in the 10 to the 8. And that's where we are right now. We really now we develop each component of the system. We're really trying to develop scalable protocol, a scale-up protocol, so we can keep the high titers all the way up to really very large scales. And that's something that's still ongoing in our lab. And this last slide is really kind of showing the importance of the system. You could take every single components by themselves, and I'm sure they're performing well, but they've really been optimized to work together. And here we've done a swap experiment in which, in some cases, we swap the cell line. For example, the LV293, those are the cells that I just showed you. Those have been developed specifically for LV productions. You could swap to over 293 cells, and you could see a big difference and big hits in the titering. Transfection region is also very important. LVR2 is the transfection region we develop in the system. A lot of people like to use PEI because it's cheaper, which is true, it's cheaper, but it's not cheaper if you look at the price per viral particles. The transfection itself is cheaper, but if the yield is 10 to 100-fold less, then at the end of the day, it's not cheaper at all. It's actually a lot more expensive to produce LV. So you got to make sure you use the right reagents. And then supplement and enhancer, you could see also they really work together to really maximize the lentiviral production. So that's why we're really pushing this whole system that have been optimized and work together and really give you the best yield possible. And so this is my last slide on the upstream here for productions. You can see the system. You can see that it's a multi-component system. It will be launched next year, but if you're interested now, it's available through beta testing program. So please contact the email highlighted here. A lot of things was testing in LV. We believe this system is now actually being adapted for AAV. There's a lot of people interested in AAV productions, and there's a little bit of a difference, but we know those cells are compatible for AAV production, so we think that can be adapted. We haven't really fully done the development there, but a lot of people are evaluating for AAV. And it's available for commercial use, meaning it's available for therapeutics. It will be a GMP products, and we could grant you the commercial rights if you want to take this to the clinic. So I'll stop right here, and then I'll send it back to Stephen that will talk to you about the downstream processing and how to purify the viral vectors, which is obviously very important as well for your application. Thank you very much.
Before turning the webinar back over to Stephen Soltis, I'd like to thank Xavier for so expertly detailing viral vector production workflows and for setting the stage for the rest of the webinar presentation. Much appreciated. If you're just now joining our webinar, thanks for tuning in. There will be a question and answer segment at the end of the presentations. Please type your question into the box on the left-hand side of your console and then hit submit. To continue on with our webinar, once again, here's Stephen Soltis. Stephen?
Although Thermo Scientific does have several solutions for purification of lentiviral vectors, such as heparin sulfate or some of our ion exchangers, I'd like to change the focus of downstream purification to another vector of gene therapy that Xavier alluded to, which is adeno-associated virus, or AAV. So purification of viral vectors, especially AAV, poses some very different challenges to large-scale processing. But before we get into that, let's go over what makes an ideal vector for gene therapy. First, the vector should be cost-effective to manufacture with a simple vector design. Now, you don't want the vector production to be overly complicated and excessively difficult to purify. And you want the vector to be highly precise. And you essentially want your gene of interest to be active only in the specific areas or tissues. Like, for instance, you wouldn't want to have a gene for hair growth being expressed in your eye. You want your vector to cause no damage to your target cell, because you don't want to elicit a strong immune response. You want it to effectively carry the desired genetic load. You want it to have low immunogenicity. And you want the capability for long-lasting expression, if that's actually necessary. And AAV fits all of these criteria fairly well. There are chromatography resins available for purifying several different serotypes. So for instance, heparin sulfate is great for AAV 2, 3, and 6. Anion and cation exchange can be used for all these serotypes. However, they bind indiscriminately to all charged particles in the feed, depending upon your ionic conditions. AAV separos is great for 1, 2, 3, and 5. But until now, there have been no really good solutions for AAV8 and AAV9 until Thermo Fisher developed the POROS, CaptureSelect, AAV8, and AAV9. So why is affinity chromatography so important in a viral production phase? Well, prior to the introduction of POROS, CaptureSelect, AAV8, and AAV9, several labs were tasked with developing processes for the purification of these serotypes. And the processes looked something like this. It would be harvest of adherent cells in either cell stacks or roller bottles, centrifugation, concentration through TFF, depth filtration, size exclusion chromatography, ultracentrifugation, ion exchange chromatography, and dye filtration prior to your final fill. So any or all of these methods could have been incorporated into their production process. And they could have actually produced a highly functional and pure product. But the problem is the typical yield with a process like this was around 10 to 20%, and typically spanned over several days and involved multiple manipulations and holds. So it's important that you understand that every single step that you incorporate into your process will diminish your final yield. Say, for instance, if you had 5 steps in your process and you had an 80% process yield from step to step, your overall yield would probably be in this range of about 20%. So, and that's not very cost effective when you're considering that you need a whole bunch of this virus in order to actually cause the therapeutic gene to work effectively. With the introduction of POROS, CaptureSelect, AAV9, one company was able to take a process with approximately a 20% yield and several steps and increase that to approximately 60% and to a 4-step process. They're also able to reduce the cost by a factor of 6. So let's talk about the resin itself. How was this affinity resin developed? Well, pictured here to the left is your typical antibody. Now, specificity towards foreign antigens is conferred through the VH and VL regions, allowing this antibody to bind a very specific recognition site. Now, there's a family of animals that produce a modified form of the IgG called the camelid Ig as 30% of their immune response, and that is what's pictured in the center of your screen. So instead of the heavy and light chain of the IgG, the camelid is comprised of the CH3 region, the CH2, and along with that is this VHH domain. Now the VHH is a 12 to 15 kilodalton molecule that confers the same specificity as a normal antibody through these complementarity defining regions, or CDRs. Now the way this resin is produced is the mRNA of the VHH directed towards a very specific binding site is then transferred and produced in yeast. And that method is an animal origin-free process. The VHH itself is very safe and well tolerated by humans. The use of this ligand immobilized on a variety of different chromatography resins has been adopted in many commercial purification processes as an affinity step. The scientists on the CaptureSelect team at Thermo Fisher Scientific have developed a ligand specifically for the purification of AAV8 and AAV9. Now, what makes the ligand directed towards the AAV8 and AAV9 particularly effective is its immobilization on the POROS resin. POROS is not your typical chromatography resin. It's actually plastic, specifically polystyrene divinylbenzene, which imparts a very robust chemical stability and makes it incompressible. What makes POROS truly unique is the pore structure on each individual bead. These hills and valleys, as you can see here up and to the left, open up a large surface area, exposing many more binding sites as well as increasing convective flow across and through the bead. When these beads are packed in a column, the structure of the bead reduces diffusional limitations, allowing for much higher flow rates. The 50-micron particle size confers excellent resolution during elution while still retaining excellent pressure flow properties. By combining the specificity of CaptureSelect and the unique structure and properties of the POROS resin, Thermo Fisher was able to create a very specific purification solution for AAV8 and AAV9 with a very high binding capacity, 10 to the 13th viral particles per mil for AAV8 and 10 to the 14th viral particles per mil for AAV9, and all with a very large range of pH stability. This resin is currently being used in several manufacturing processes. Genethon shared with us some of their process data. So they previously used a 3-step ion exchange method, which they switched to a 1-step affinity process and achieved the same degree of purity and improved their yield to greater than 80% recovery. More importantly, the process yield was consistent among several different batches. So it's important to note that the resins are not simply plug and play. The purification of AAV poses a very unique challenge. So AAV replicates in the nucleus of the cell and is not released until the cell is disrupted or undergoes apoptosis, which causes the release of several different contaminants. So let's imagine these beads in a chromatography column. So the tropism of the viral capsid, especially AAV9, is directed to many different cellular proteins. And these proteins will definitely be contaminants. So when AAV binds the resin at first, there's typically enough distance between the particles, allowing the contaminants to flow through freely. As binding progresses, so much AAV binds to the resin, essentially forming a charged surface for contaminants to bind to. Thus, these bound contaminants could elute along with the virus. And these contaminants include host cell proteins, host cell DNA, plasmid DNA, media components, transfection reagents, additives, and even helper viruses if you choose to use them. You typically eliminate these contaminants by incorporating a secondary wash. So you can use 1 molar NaCl at various pHs, magnesium chloride, chaotropic salts, arginine, ethanol, but in this case phosphate buffers should be avoided, detergents, glycol, and organics. You can also optimize your elution strategy to maximize your recovery by employing the following strategy. If you start with 100 millimolar citrate pH 3, and then measure your viral yield in your feed, as well as various points in your process, you can determine if your elution is fairly efficient. And then you can start adding these additives to maximize your recovery, such as phosphate, glycine, magnesium chloride, propylene glycol, or arginine. So it's important to note that all processes are different and could require none or several of these additives to maximize your recovery. As mentioned earlier, POROS has excellent pressure flow properties. So you will be able to flow at flow rates greater than that of a typical soft gel resin, yet still retain a decent binding capacity. This will allow you to shorten your process times, possibly allowing your manufacturing team to purify a batch within one working shift. Speaking of batches, this resin is very scalable and reproducible. As seen in this graph from Genathon, they were able to achieve reproducible results from 10 to 50 to 200 liters. So you may be wondering about empty-full ratios, or at least the polishing step after affinity. This step is typically done using ion exchange. Both anion exchange and cation exchange can be used for AAV binding. This is the way it works. If an AAV particle is exposed to higher pH, the surface proteins displayed have an overall negative charge, allowing you to use anion exchange to purify it. The same goes if you expose AAV to lower pH conditions. In this case, the surface proteins displayed have an overall positive charge, allowing you to use cation exchange. So empty capsids are considered a contaminant, and they occur as a byproduct of AAV synthesis. The capsid is typically formed first, and then the DNA helicates into the capsid, forming the full AAV particle. The sizes stay the same. However, the density is different, allowing for ultracentrifugation, which is not very scalable. There's also a difference in PI, and the difference in PI means that you can exploit the difference in charge by altering the elution conditions using ion exchange chromatography. So here we see an instance where one of our anion exchangers, HQ, was used by this group to separate empty and full particles. In this case, AAV2 empty and full particles were loaded onto HQ. The slight difference in charge at particular pH conditions allowed these researchers to manipulate the elution to effectively separate empty and full particles. And then when the researchers ran the individual peaks back over the column, a single peak was formed, indicating that efficient separation of empty and full occurred. So Thermo Scientific offers several anion exchangers, XQ, HQ, D, and PI, that are all on the POROS backbone. Although the charge characteristics are slightly different for each resin, you can expect the same resolution, binding capacity, and flow characteristics you would see with any POROS resin. Again, all processes, and in that case serotypes, are different, so it is very important that you try all of these ion exchangers in your process and see which one works best for you. So we offer solutions for both affinity and polish of AAV particles. Affinity resins offer high selectivity, capacity, and scalability in the purification of these vectors for the production of clinical-grade gene therapy molecules. These resins can significantly improve your downstream processing by reducing the number of your purification steps. And it's all backed by state-of-the-art manufacturing and a technical support team. So now I'm going to go into a little bit of the characterization and analytics of viral gene therapy particles. Now I'll speak to AAV again because, well, that's what I know best. So what you'll see here are some of the assays that most of us in the gene therapy world are very familiar with. So silver stains for purity, qPCR for dot blots and titering, alkaline gels for packaged genome, and electron microscopy for the empty-full ratio. And Thermo Fisher offers solutions for all of these different tests, including various staining kits, the PCR system, the eGel imager, and we even offer an electron microscope. We also have several offerings for leakage ELISAs to test for leachable ligands when using the Affinity resins, and HPLC columns, which can be used for titer determination. We also offer conjugated ligands, which are biotinylated for all your immunoassay work. Now, there are several other assays that must be performed, and Thermo Scientific has devoted an entire division for these analyses called Pharma Analytic. The Pharma Analytics group develops rapid molecular methods for pharmaceutical manufacturing. These solutions are designed to meet or exceed the regulatory requirements for the industry. The product portfolio is built upon DNA sequencing and real-time qPCR technologies. The methods that are applicable to gene therapy manufacturing include bacterial and fungal identification, viral and mycoplasma detection, and residual DNA quantification. The ResDNA-seq residual DNA quantitation system is a premier quantitation system for residual DNA. The method's high sensitivity, high specificity, and consistent performance generates accurate, reliable, and reproducible data that enables improved process understanding. The system includes a dedicated software solution to help with 21 CFR compliance. A second qPCR assay delivers a presence-absence call for mycoplasma in manufacturing samples. The MycoSeq Mycoplasma Detection system provides a rapid 5-hour solution for mycoplasma detection as an alternative to the traditional 28-day growth test, which typically delivers a result only after 30 to 40 days. This qPCR-based method uses a proprietary set of oligonucleotides to detect more than 90 mycoplasma species with a sensitivity of less than 10 copies per reaction, which is required by regulatory authorities for lot release testing. The easy sample prep also supports in-process testing. The MicroSeq system uses DNA sequencing for microbial identification. Genotypic data, as an alternative to traditional phenotypic data, provides the right answer the first time for cost-effective testing. As an established platform in the pharmaceutical industry, 24 of the top 25 pharma companies trust the MicroSeq system for microbial monitoring and identification. Lastly, the ResDNA-seq, the MycoSeq, and ViralSeq assays all leverage a set of platform instruments to provide a fully integrated solution for a broad spectrum of contaminants and impurities. This includes an Automate Express for automated sample preparation, reagent consumables, the 7500 qPCR instrument, and the AccuSeq software for rapid data analysis, with features that help with 21 CFR compliance. So we have presented solutions offered by Thermo Scientific for upstream and downstream purification, including cell culture offerings, purification through chromatography resins, and analytical solutions for the clinical manufacturing of gene therapy vectors. We help accelerate innovation and enhance productivity for our customers. With every decision, we ask, will this accelerate our customers' ability to innovate? Will the productivity in their lab be more improved or enhanced? Our customers are at the center of everything we do. The products and services are very important, but the actual benefits they offer to our customers are what ultimately allow us to deliver on our commitment and our value proposition. So with that, I thank you for attending this webinar, and we will open the floor up for questions.
Thank you, Stephen, and thanks again to you, Xavier, for your in-depth discussions on Thermo Fisher Scientific's approach to providing platform solutions for viral vectors. Your presentations definitely caught the attention of our audience based on the number of questions we received. So please bear with us now just for a few seconds as we get set up for the Q&A portion of our webinar. Okay, so Xavier, we have the first question. Let me pull that up.
Okay.
Okay. So, I wanted to ask you if your enhancers are based on serum-free and animal-free components.
Yes, they are. And I didn't fully mention, but the system, cells, and media is all serum-free.
Thank you. And will you be able to provide cells with GMP-grade documentation and allow vendors to use this cell line for GMP production?
Absolutely. That's the plan. That's really the plan of this product and this program, is to allow people to be able to produce LV for clinical therapeutic use. So all the products are GMP, including the cells, and the cells will come with documentations for that purpose.
And Xavier, what is the suspension 293S?
They are specialized cells derived from 293Ts, specifically designed to maintain high titers, high density, and high production of LV. But the origin is 293Ts.
Okay, and I have another question. Generation 1, 2, 3, and 4, are they different generation LVs?
Yes. So, okay, sorry. So maybe I wasn't confusing. I was confusing a little bit. The vectors is the same. The vectors we use is viral power, lentiviral. What generation 0, 1, 2, 3, 4 means is the iterations of the system, meaning better media, better cells, optimization of the protocol. It's kind of the evolution of the program. So generation 4 is kind of our optimized system with all the components. But the vectors stay the same all along.
And Xavier, is the beta testing doable with AAVs?
Yes. So beta testing is really it's kind of putting our product out there in the hands of our customers. So obviously we do mostly the design and the developmental work with LV, but there's definitely a lot of interest in people want to try for AAV. So it's absolutely possible to try for AAV. We just don't have as many data as we have for LV. So I'm personally interested for those kind of beta testing, so we can work together and see if the system works well for AAV.
And Xavier, which PEI have you used in that experiment?
I believe we used PEI Max in this experiment. There's a lot of different PEI with a lot of different performance. I think PEI would be the less worst of all the PEI, and we use PEI Max.
Okay, Stephen, we did not forget you. What preparative procedures should be performed prior to loading onto the Affinity column?
It's important that you, of course, get as much AAV out of the process as possible, so you can actually do anything you want up to that point. It's important, however, that right before you load onto the Affinity column that you 0.2 micron filter. That is the only recommendation that we have.
Okay, Stephen. And are there any cases where the cells do not have to be ruptured for AAV purification?
Sure. There was a paper published not too long ago that said that AAV9 actually ended up with 90% in the media, in which case then if you have that sort of process or that sort of upscale process, then what you could actually do is start directly from the media. However, you still have to 0.2 micron filter your feed prior to loading onto the Affinity column.
Okay, thank you. And back to you, Xavier. Do you know if there is a benefit to using lipofectamine over PEI for upstream AAV production?
Yes, absolutely. I mean, the benefit is this is a transient transfection, so you're trying to transfect a plasmid into those cells, and actually multiple plasmid. Lipofectamine products will give you simply better transfections. Better delivery of the DNA, better expressions will result in a lot better LV productions. So there's no doubt there's definitely an advantage of using better transfection reagents like lipofectamine.
And Stephen, what are the guidelines for empty-full ratios?
To be honest, and to answer the question fairly quickly, there are none. It's just important that when you go to test your vectors that they remain consistent from prep to prep.
Okay, and Stephen, do you offer an affinity resin for a lentiviral purification?
Yes, we do. Not only does we offer, well, actually, it's in development. So our team right now at CaptureSelect is developing a VSVG-specific lentiviral vector affinity purification resin. But on top of that, heparin sulfate actually works fairly well for lentivirus purification.
And Stephen, separation of empty and full AAV particles is still a major issue in AAV production. Has IX-based separation of full empty ever been demonstrated?
Sure, other than AAV2. Yes, so it's actually been shown for AAV1 and AAV8 using anion exchange chromatography. And in the case of AAV1, there's a paper published where they're using the HQ resin available by Thermo Fisher.
And Stephen, are the POROS columns compatible with non-Thermo Fisher purification systems such as ACTA?
Oh, absolutely. They're fully compatible with any chromatography system that you want to use. And in cases where you don't know how, we'll be happy to show you how.
Okay, well, thank you. Well, unfortunately, we've run out of time, but we would very much appreciate it if you would answer a short survey by clicking on the small green widget on the lower right of your computer screen. We thank you very much for that. Also, please note that this webinar will be archived for 1 year on our website, www.genengnews.com. If you miss parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. Thanks again to the panel for your outstanding presentations, and I want to express our appreciation to our audience for your attention and for your very thoughtful questions about the specific topics brought up during the webinar. And thank you to Thermo Fisher Scientific, whose support made this webinar possible. Bye-bye for now.